I would like to start with this quote by Amy Maxman, as it truly characterizes the power and potential of genome editing. The first example of genome editing was in 1972 when Paul Berg, a biochemist from Stanford University, produced the first molecule of recombinant DNA. Later that decade, in 1976, Herbert Boyer and Stanley Cohen filed a patent for the first recombinant DNA-based technology, and this was granted in 1980. Boyer then went on to co-found Genentech, which which came up with the method of mass-producing insulin with bacteria that had recombinant DNA that allowed them to produce insulin in rates that had never been seen before. So the development of modern genome editing techniques. In my opinion, the very first example of genome editing was selective breeding practices. These were carried out in the agricultural revolution of the 1800s, and this was the first time that the genome of animals had been not specifically altered, but had been changed by humans, as in if they were left to breed naturally, it would have been different to how they were bred, as they were as the two parents were chosen specifically to breed for specific phenotypes. Next, in 1987, a professor named Yoshizumi Ishino mentioned CRISPR-based technologies in an academic paper in 1987. Jennifer Doudna, who is known as the mother of modern CRISPR and genome editing, started working on CRISPR in 2005, and she theorized that it could be used as a programmable genome editor. Spoiler alert, she was correct. In 2013, CRISPR was first used to edit a human genome. The discovery was actually made in late 2012, but wasn't put into practice till early January 2013. Now, more recently, prime editing, a more reliable genome editing technique that's based on a similar mechanism to CRISPR, was published by MIT, and this happened in October 2019. So actually very recent, October 21st, 2019. And due to its relative infancy as a genome editing method, it's still not being used in commercial settings. The paper that was published on the 21st of October is actually called "Search and Replace Genome Editing Without Double-Stranded Breaks or Donor DNA." You will get into what that means later on, but real quick, double-stranded breaks, or DSBs for short, are the result of CRISPR, as in CRISPR induces double-stranded breaks, which allows new DNA to be placed into the genome. The head author was A.V. Anzalone, and it was published in Nature, the Nature journal. And finally, the most recent sort of development of genome editing was in 2020 March, so very recent indeed. A patient received a CRISPR-based therapy directly into their body. Now, we don't know if this was successful or not, and we, as part of the biotechnology community, we all hope that it's a successful case, and hopefully the start of many successful therapies using CRISPR and genome editing technologies. The genome editing mechanism. Now, most genome editing mechanisms work in a similar way, and the biggest one is CRISPR-Cas9. Now, the mechanism actually was found as an immunoresponse in the Streptococcus pyogenes M1 bacteria. It would produce the Cas9 nuclease enzyme, which means it's designed to break down DNA and genomes. It would produce this enzyme along with the gRNA when it was infected by a virus. Now, a virus would infect it by inserting its own DNA into the bacteria. Now, the bacteria would produce this gRNA, which was an exact replica of the viral DNA. That means that, and this would bind to the Cas9 to form the Cas9 complex, which is shown here. Now, this Cas9 complex, because the gRNA is an exact match to the viral DNA, can find the viral DNA and bind to it. The gRNA means that the complex can only bind to a portion of DNA that is the exact match, which stops the Cas9 from binding to the bacteria's own DNA plasmids that could cause damage to the bacteria's own DNA. So this complex then binds onto the viral DNA, and it binds at the PAM, which stands for the protospacer adjacent motif, which is sort of like a promoter sequence, and this allows the Cas9 so once the gRNA finds the PAM and binds to it, it allows the Cas9 complex to start cutting the DNA. So in a in a bacteria cell, this is a good thing, as this means the viral DNA is now destroyed. But as you can use this technology in vivo to edit live genomes, it creates a DSB, which is a double-stranded break, which then allows you to put in a new strand, a new sequence of DNA, and this allows you to edit their genome. So if you're putting in a new strand of DNA, it's an insertion, or you can simply delete DNA, and then an insertion can sometimes also cause a frame shift, and this is how the genome editing mechanism works. Now, there are many applications, especially more that can be discovered in the future, but even now, there are many applications for genome editing mechanisms. But today we'll only discuss the ones that I've chosen to be the most interesting and the most revolutionary, in my opinion. First is the treatments for HPV, which is a hepatitis B virus, and this virus is relatively invulnerable to modern day antiviral therapies, as the virus contains covalently closed DNA, cccDNA, which means it cannot be targeted by normal antiviral therapies. But the CRISPR-Cas9 endonuclease complex can be used to induce the DSBs, or the double-stranded breaks, within the cccDNA of the virus, destroying it and making it redundant, therefore not able to damage its host. This is able to be done because of the gRNA that is within the Cas9 complex that allows it to target a very specific sequence of base pairs. Next, we have stem cell treatments for muscular dystrophy. Now, TALENs is another form of genome editing, and it stands for transcription activator like effector nucleases, and it's a part of the family of nucleases, which means enzymes that are able to destroy or break down DNA. And TALENs is used to correct the frame shift mutation that is responsible for muscular dystrophy, and this is done within stem cells that can be put back into the affected patient. And as there has a unique target region, the TALEN is very unlikely to bind to any other site within the genome, within the stem cells, making it very efficient and relatively safe. Next, we have Zetafen treatments for HIV. Now, Zetafen is the third of the large genome editing mechanisms that are used today, and Zetafen stands for zinc finger nucleases. Similar to the Cas9 nuclease, breaks down DNA and is used to break down or to remove the CCR5 gene, which codes for a protein which acts as the main receptor for the HIV virus. So the Zetafen treatments are very effective in HIV, in treating HIV, and a study done by Tebas, a scientist with a last name Tebas, and he wrote a paper with his whole team called Gene Editing of CCR5 in Autologous CD4 T cells of persons infected with HIV in 2014. And in that paper, he details how the 1 in 4 patients reached undetectable levels of viral load within their blood, and most, if not all of them, showed a large decrease in viral load, meaning it's a very effective treatment for HIV. The last medical application we'll be talking about today is the treatment and screening for cancer, and this is done by targeting the BRCA1 gene, which is a tumor suppressor gene. So inserting this gene can make those who are vulnerable to cancer less vulnerable, and this can also be used to screen for people who have cancer. Finally, the last application is for research, and CRISPR can be used to visualize genomic loci, which means sort of the architecture of the genome and how exons and introns fit together and how they have an effect on the proteins and the phenotypes that they code for. And this research can only be done by visualizing the genome, and the most efficient and best way to visualize it is using CRISPR. So the ethical implications of genome editing mechanisms, especially if they were to go mainstream in the future. Now, the biggest concern, or concern that a lot of people have, is possible the frivolous usage, such as its use for cosmetic changes, as it is a possibly dangerous procedure. So it could be used like plastic surgery and therefore used for purely frivolous cosmetic reasons. Uh, similar argument behind the abortion, whether abortion should be illegal or not, such as when you're editing the genome of an embryo, you're doing it without its consent. So whether a parent should be allowed to choose whether the genome of its future child should be edited or not. Accessibility, those who need it might not be able to get it either due to monetary reasons or legal reasons, and this would be a large travesty. Finally, uh, the hindrance to those who need it. So those who are born with genetic diseases may not be able to get it because of monetary or legal reasons. I would like to finish with this quote, as I feel that this truly characterizes the problem of genetic diseases that lies before us today. And it is my strong belief that genome editing mechanisms is the future for solving these diseases, and its capacity and potential far surpasses simply curing these 6,000 or more genetic disorders, to elongating the human lifespan, to things we can't even imagine yet. And it all stemmed from something as simple as the bacterial immune response for viral infection from the Streptococcus pyogenes M1 bacteria. And that's something that I find truly amazing. Thank you for listening.